everyone, I am Dr. Sonal Jain from Department of Anthropology, University of Delhi. In this series of forensic science lecture, today we are going to learn about centrifugation. Centrifugation is a very important technique which is used in instrumental techniques when we have to analyze any forensic chemicals or any forensic biological material. Centrifugation is one of the important technique where we have to separate certain materials. It is combined of various other techniques depending upon what kind of material we have to separate into different parts. We go for centrifugation. To start with, we say the centrifuge is a device which is used for separating particles from a solution according to their size, shape, density, viscosity of the medium. Basically, the centrifuge which we are talking about, it uses centrifugal force to separate the faces of different densities. That means it is using the density of the material as a way to separate it where we are applying the centrifugal force. The centrifugal force is proportional to the rotation rate of the rotor. Rotor is in the center and which is used for the rotation. It is basically consists of a rotor which is enclosed in a refrigerated chamber and which is spun by an electric motor. So, whenever we discuss or talk about centrifugation, there are two terms we need to learn. One is sedimentation and one is centrifugation. When we say sedimentation, when a suspension is allowed to stand, the denser solids slowly settle under the influence of gravity. So, that is sedimentation. The principle of sedimentation is used in centrifugation. It is a settling process that is accelerated with a centrifugal field. So, we know that the particles or the materials which have a higher density, they tend to settle down because of the gravity. So, when these particles they are settling down inside the tube, we also put a centrifugal force around it which rapid up this process of settling down. One of the important example which I can give about the centrifugation which we use in forensic science is the extraction of any biological material. For example, the extraction of DNA or RNA or separation of proteins, we use centrifugation technique at each and every step. For example, if we have to go from blood to DNA, we have to cross many walls, many cell walls, many different bodies are there which we have to separate out of the blood, and then we have to reach up to the nucleus, and after breaking the wall of the nucleus we have to get the DNA out of it. So, all these processes include sedimentation and centrifugation. It is based on this that different materials which are present in the blood, they have different size, shape, density, and they have different sedimentation rate. So, on that basis we can use sedimentation, that is the principle, and the technique of the centrifugation where we use the centrifugal field which can help us in the settling down and looking at the particle which we are more interested in. So, when we talk about the centrifugation, basically broadly we say it is of two types. One is analytical and one is preparative. Analytical, as the name suggests, it involves measuring the physical properties of the sedimenting particles such as the sedimentation coefficient or molecular weight. So, mostly when we are going for the analytical centrifugation, we usually put a microscope and a detector in the centrifuge and we see how the particles are behaving, what is their sedimenting coefficient, what is their molecular weight, and when we are providing the centrifugal force, the behavior of particle is being studied in the analytical centrifugation. Basically, we include other techniques also to know the behavior of the particles, to study their sedimenting behavior. Then moving on to the preparatory centrifugation, the objective of this is to isolate specific particles that can be reused. For example, let us talk about the extraction of DNA. So, if we are extracting DNA out of the blood, we need the purity of the DNA because we have to create further studies on the DNA which is extracted using this process of centrifugation. So, basically different buffers are used at different steps and in the end result we get DNA out of the preparative centrifugation technique. As we say, there are many types of preparative centrifugation. For example, rate zonal, differential, isopycnic centrifugation. All this technique, they based on simple principle of centrifuge and centrifugal force. So, centrifuge is a piece of equipment which is generally driven by an electric motor that puts an object in rotation around a fixed axis, applying a force perpendicular to the axis and to separate substances of different densities. So, what did we get out of this? That whenever there is a mixture and we have to separate the mixture and the different particles which are present there having different densities, they can be separated using the centrifugal force. So, whenever we are using a centrifuge, the tubes in the centrifuge either they could be straight or they are tilted so that the centrifugal force can pull denser substances towards the bottom of the tube. In certain cases, as we have observed in the forensic biology, the main important material when we extract the DNA from the various biological materials found at the crime scene, this is a very important technique, and without the use of centrifuge or the process of the sedimentation, it is not possible to isolate DNA out of the various biological materials. As we can say, whenever we are using the principle of sedimentation and centrifugation, there is a deposition below the tube where we have two different types, the pallet, and then we can remove the supernatant or we can use the pallet, whatever we are looking for. So, the important thing where the centrifuge works upon is RCF. Now, what is RCF? It is the relative centrifugal force. It measures the acceleration applied to the sample. So, as you see in the diagram that we note the radius from the center till where our sample is placed. RCF, when we say, it is R multiplied by W square divided by G, where R is the distance from the center of the motor to the sample, W is the angular velocity which is being measured in radians per second, and G is the force of gravity which is 9.8 meter per second square. So, depending upon different types of materials, they require different speed, they require different centrifugal force to get separated. So, as we see, as we take the example of whenever we are studying the centrifugation, we take step by step the example of the extraction of DNA. At different points, when we know that we have to break the wall of WBCs, or we have to break the wall of the nucleus, or we have to separate the other material out of that. So, depending upon different buffers are used, and depending upon that different speed is used, so that we can isolate the DNA out of the blood or any other biological material which is found at the crime scene. And we use the basic formula of RCF is equal to R into W square by G. Now, coming to the types of centrifuge, we depending on the particular application, centrifuges differ in their overall design and size. A common feature which is present in all centrifuges is the central motor. This central motor is present in between the instrument and that spins a rotor containing the sample which is to be separated. So, basically whenever we are using any centrifuge, the motor will be in between and the rotor will be there on the outside of that, and in the rotor we will keep our samples. So, when we talk about centrifugation, there are two terms which we have to keep in mind. One is supernatant and one is pellet. So, while we were talking about the sedimentation, we talk about that different materials, they settle together at different rates depending upon their density. Whenever we have a mixture of different substances having different size, shape, particle size, densities, a particular centrifugal force is applied to them. The heavier particles with a higher density, will settle down in the tube. We call that as pellet, and the remaining material, it is suspended over the pellet, and that material is called as the supernatant. As you can see in the image, the below material is pellet and the upper material is supernatant. If we require something from the pellet, then we discard the supernatant and we can use the pellet, and we can go ahead using centrifugation at various steps. If we require the supernatant and we have to throw the pellet, we can take the supernatant in the different tube and then we can go ahead with our further analysis. So, any extraction we are doing, for example, of the DNA from any biological material, we must know that at which step we are looking for what kind of cells will be found in the pellet or supernatant. Is supernatant is of our use, or if it pellet is of our use. So, we have to take care depending upon the sedimentation rates of different particle size which we are looking for. So, now this is how a centrifuge looks like. Depending upon what kind of centrifuge is that, what kind of tubes can be used in. For example, there are microcentrifuge where we use the micro tubes having the volume of 2 microliter, having the volume of 200 microliter, 500 microliter or 2 ml, or we have the rotors where we can use of tubes of 10 ml, or where we can use the tubes of 50 ml or 100 ml. Depending upon the centrifuge, the type varies and the amount of blood we are using for the extraction of DNA or any other thing. For example, for separation of certain particles, we use the centrifuge. Depending upon how much material we are starting with, 50 ml, 10 ml, 5 ml, 2 ml, 1 ml, etc. The rotor can be changed as per. The rotor, depending upon the size of the tube, we can put a different rotor. It depends upon machine to machine that whether it has a changeable rotor machine or a machine with a non-changeable rotor. So, the centrifuge depends upon various factors like the maximum speed of sedimentation, whether the vacuum is present or absent, whether the refrigeration is a temperature controlled refrigeration, and the volume of sample and capacity of centrifugation tubes. So, if we have to extract the DNA out of 10 ml blood, this rotor cannot be used because this is for 2 ml tubes. We have to replace it with a 10 ml tube rotor. Depending upon different types, the first one is small benchtop centrifuge. This comes with or without refrigeration. There are many companies which manufacture centrifuges, many Indian companies, many foreign countries. Depending upon company to company, their centrifuge come with or without refrigeration. Now, what is the necessity of refrigeration in the centrifuge? It is that whenever we are extracting something, whenever we are separating something, whenever we are purifying something, we require certain temperature at a certain point of time. This refrigeration will provide a specific temperature which will help us in the purification or the separation of the substance. It has slow speed, for example, up to only 4000 RPM. What is RPM? It is revolutions per minute. That means how many times the centrifuge is revolving around the motor in a minute. This type of setups is commonly available in clinical labs where we have to separate blood, plasma or serum. It can take up to around 100 tubes depending upon the diameter of a small benchtop type centrifuge. Now, the next one is the microcentrifuge. This is the most commonly used microcentrifuge for the extraction of DNA and RNA, because mostly whenever we are extracting the DNA, we are taking up to 1 ml or 2 ml of blood for the extraction of DNA. This is known as a microcentrifuge because The tubes which are being used inside this are the microfuges or append of small tubes. They have the volume of up to around 2 ml. This is very common in biochemistry, molecular biology and biological labs, and this can generate forces up to 15,000 G. G is the measure where we are using the force of the centrifuge. This can be with or without refrigeration. Mostly microcentrifuges which are available in the biological labs for the extraction of DNA, we use the centrifuges which are refrigerated. Now, the third type is high speed centrifuges. When we say the high speed, then it says that the speed is around 15,000 to 20,000 revolutions per minute. The centrifugal field is around 1 lakh G. A large sample capacity depending upon the rotor. Normally the high speed centrifuges are refrigerated and they have research applications like differentiation, separation of nucleus, mitochondria, protein precipitation, large intact organelles extraction, cellular debris, bulky protein aggregates. So, basically whenever we are doing the molecular biology work, we require the high speed centrifuges. Now, the next category is ultra centrifuges. Why it is ultra? They have the highest speed which can be achieved, that is 65,000 revolutions per minute, and which has around lakhs of centrifugal force. But the basic point is it is very expensive, it has a limited lifetime and it requires very special rotors to operate upon. For the care, the balance is critical in each and every centrifuge, and it has much research applications when we talk about also the purity of different molecules. When we talk about the ultra centrifuges, the high speeds used in such devices, they generate a very high amount of heat. Therefore, we have to take proper measures for cooling arrangements, and vacuum is required in these kind of ultra centrifuges. Then we have large capacity preparative centrifuges which have centrifugal field around 3000 to 7000 G. This is for the efficient separation of coarse precipitates or whole cells. The low speed centrifuge, they are mostly used in the laboratory where routine sedimentation of heavy particles is required. They have a maximum speed of 4000 to 5000 revolutions per minute. Usually when we talk about the low speed centrifuges, they operate at room temperature and they do not require any temperature control. There we can have two types of rotors, one is fixed angle rotor and one is swinging bucket rotor. We will discuss these type of rotors further in our presentation. It is used for the sedimentation of RBCs until the particles are tightly packed into a pallet and supernatant is separated by decantation. So, as we previously said, we must know whether we are interested in the supernatant or the pallet. So, if we are interested in the supernatant, we decant it and then we can use it in the another tube. If we are interested in the pallet, then we can throw the supernatant and we can go ahead with the pallet in the same tube. So, the tabletop centrifuge, they have different type of rotor, either they have fixed angle or they have swinging bucket type of rotor. When we talk about the high speed centrifugation, they are used in high sophisticated biochemical applications where we require higher speeds, higher temperature control, then the rotor chambers are essential. The operator of these instruments can carefully control speed and temperature, which is required for very sensitive biological samples. Where we have to extract a particular or purify certain biological applications, then we use high speed centrifugation. Usually when we talk about the rotors which are involved in high speed centrifugation, they are fixed angle, swinging bucket and vertical rotors. So, they can be, if the rotor is at an angle, it can be a fixed angle, and if it is not an angle, not fixed, then we call that as a swinging bucket. Ultracentrifuge is the most sophisticated instrument. In this type of centrifuge, intense heat is generated due to high speed. Thus, the spinning chambers must be refrigerated and it should be kept at very high vacuum. These ultracentrifuge can be used both in preparative centrifuges as well as in the analytical work. So, if it is used for preparative work, then we call it as preparative ultracentrifuge, and if it is used for analytical work, we call it as analytical ultracentrifuge. They are composed of 4 parts: drive and speed control, temperature control, vacuum system and rotors. So, drive, water cooled electric motor is there. For speed control, it is selected by rheostat, monitored with a tachometer. When we talk about the overspeed system, we should prevent the operation of a rotor above its maximum rated speed. So, supposedly if they say that this centrifuge can only be operated till 12,000 rpm, we should never operate it above 12,000 rpm. Whatever the maximum rated speed is mentioned in that particular manual, we should always operate it on that particular speed. Otherwise, the centrifuge can go bad. It consists of a ring of alternating reflecting and non-reflecting surfaces which is attached to the bottom of the rotor. This is a small but intense point source of light and it contains a photocell. But when we talk about the temperature control in the high speed centrifuge, we usually place a thermocouple in the rotor chamber, monitor only the rotor chamber temperature, whereas in the ultracentrifuge, an infrared radiometric sensor is placed beneath the motor which continuously monitors the rotor temperature. Why the temperature control is required? Because whenever we are reaching such high speeds of such RPMs, heat is being produced. So, we have to take care of the temperature control in such cases. So, when we say the rotor of 2 types, mostly in the low speed centrifuge, they are of 2 types: angle rotor and swinging bucket rotor. Angle rotor, they are consist of a solid piece of metal with 6 to 12 holes. Depending upon what kind of company rotor we are using, we use an angle rotor or we use a swinging bucket rotor. The angle usually lies between 20 degrees to 45 degrees. Whereas swinging bucket rotor, we have hanging 3 to 6 free moving buckets. So, as in the first image we can see, this is a swinging bucket rotor where the buckets are hanging, and when the centrifugal field is applied and the force is applied to this rotor, the buckets, they will go in the perpendicular position up to 90 degree, and then the sedimentation will happen in the tube when it reaches the 90 degree. Depending upon, when we say, the most important thing in the centrifugation is the balancing. As you can see in the first image where we have written 2, if we have 2 tubes and it is a 6 hole centrifuge rotor, then we put them opposite diagonally. In a 6 tube system, if we have 3 tubes, we can put them in alternate. When we have 4 tubes in a 6 one, then we can put the equal quantity opposite to each other. Similarly, in the 6 type rotor, we put equal quantity opposite to each other. Balancing is the key in centrifugation. When we talk about the centrifuge rotors, the fixed angle rotors, as you can see in the image that it is at a fixed angle as we spoke, which is around 20 degree to 45 degrees. Sedimenting particles have only short distance to travel before pelleting, shorter run time and the most widely used rotor type. Whereas in the swinging bucket, the longer distance of travel may allow better separation, easier to withdraw supernatant without disturbing the pallet. So, depending upon what kind of rotor we are using, what kind of separation is required, what kind of material we have to extract or separate, we use the rotor based on that. So, just to conclude, I will say the centrifugation is one of the most important separation technique which is being used in forensic science. Thank you.